आइए हम सुने विज्ञान की अनकही कहानियाँ। आइए हम सुने विज्ञान की अनसुनी कहानियाँ। एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़।
Celebrating Azadi Ka Amrit Mahotsav, we bring you a podcast series on STEM popularization, science, technology, engineering, mathematics and medicine, supported and catalyzed by the National Council for Science and Technology Communication, Government of India. This program is produced by Media for Community Foundation. Creative content and technical support is given by Purvasha Banerjee and Shushanto Das.
एक नई दुनिया की आवाज़ आपकी आवाज़, विज्ञान की आवाज़, की आवाज़, की आवाज़।
Vigyan Ki Awaaz podcast series aims to popularize new ideas of science and technology, which highlight STEM, science, technology, engineering, mathematics, and medicine. This brings to the surface the efforts and researches of Indian scientists, which is desired to bring a change in community learning and teaching, access to knowledge of resources, and social paradigms. Our goal is to spread the name of indigenous Indian research stories and their masters among the masses of society in a very easy manner. Bose Institute was set up in 1917 by Sir Jagadish Chandra Bose, the founder of modern science in the Indian subcontinent. It is Asia's first modern research center devoted to interdisciplinary research and bears a century-old tradition. The institute was founded as not merely a laboratory but a temple, with its principal aims being the advancement of science and diffusion of knowledge. Swami Vivekananda, Sister Nivedita, whose influence is reflected in the institute's emblem of Vajra, was significant in the setting up of the institute, and Rabindranath Tagore were among those who had consistently inspired and supported Jagadish Chandra Bose in his endeavors. Ever since the establishment of the Department of Science and Technology, Government of India in May 1971, Bose Institute has been an autonomous grant-in-aid institution of DST. Over the last century, the institute has endlessly strived to preserve the legacy of its illustrious founder. We have Professor Pallob Kundu in this episode, who is a professor at Bose Institute. He shares about his journey as a researcher and about his current research.
I am Pallob Kundu. I am currently working as a professor at Bose Institute. So I have 30 years of research experience in specifically in molecular biology field. Because I started my PhD at Bose Institute and then I moved to USA for doing my postdoc. So during this whole process, I worked on specifically on molecular biology and different aspects of molecular biology. So currently my research is on plant molecular biology and particularly research interest is on improvement of crop plants. Now I have basics study I did with BSc Agriculture. And then I did my master's in biotechnology from Madurai Kamraj University. So that's why I have basic interest in crop improvement via biotechnology. And when I joined Bose Institute, so I selected the topic of improving crop plants via some genetic engineering technologies. But prior to that, we need to understand why do we have to do the genetic engineering. So if you just go outside and look at many plants, but you will not find that many plants are infected actually. So disease is not that common for general plant. However, if you just go to any cultivation field, you'll see that many plants get infected, and to prevent that, you know that farmers are usually spraying tons of pesticides, fungicides and insecticides. So those are not good. So to avoid that, so best method of improving this crop health, let's say, or to avoid that using this pesticide, so we can boost a plant's immune system, own immune system. So then how do we boost a plant's immune system? To boost the immune system, we have to understand that how plant resists the diseases. So my research topic is one of the aspects of how plant can resist the diseases. So if we just try to understand that how plant can resist disease, then obviously there are different aspects of this plant resistance at the molecular level. But in general, when the plant is infected, what plant tries to do is that restrict the pathogen by killing those cells where it is infected. So if the cells are killed, plant pathogen can get arrested in that particular place. But to do that, so plant usually activate several molecular pathways, and we are looking at that how these molecular pathways are getting activated and what are the regulators of those pathways. So there are many molecular regulators, that's where our interest is. Now if we can understand that how these molecular regulators are working during pathogen, so probably then we can manipulate them by genetic engineering and then bring the resistance. So that's just my research topic. And the plant we work with, the tomato plant, and pathogen, various types of pathogens like some fungal pathogen which causes the early blight disease of the tomato, or other pathogens like Pseudomonas and Xanthomonas pathogens. So all down, then why do we work with the tomato plant? You know that the tomato plants are useful. We all eat tomatoes, and it's an important vegetable crop, and it's a very good model system for studying plant pathogen interaction. So that's why we are focusing on that. And also another aspect is that tomato plant genome is very well understood, sequences are known like that. So as I told you earlier that we are working on certain aspects of how plant is resisting itself. So now we have identified certain pathways, molecular pathways, which are important, which are getting activated during plant pathogen interaction. Now via genetic engineering, we are trying to modify those pathways and to bring the resistance. And for that purpose, one common technique we are using, which is newly developed techniques, is called gene editing technology. So via gene editing technologies, we are trying to modify certain genes and then try to improve the plant. Now, if you think that then where is this research is standing in Indian context? Yeah, in India, different laboratories are trying in the similar techniques. However, we are using certain new technology we have developed for gene editing. So those technologies we are using to modify genes as and when plant will get infected. It's not that always it will be activated or gene will be nullified, but when it will be required, so only then it will be activated or deactivated the gene so that plant can resist the pathogen successfully. So there are different industries in USA, let's say. They have developed certain genetically modified crops, but you know that genetically modified crops are not allowed in India. But it does not mean that we cannot do it now. So our motto is to we get ready. Like we know that which genes has to be modified, which pathways can be altered, and if we have this technology ready, so when let's say that government policies will allow using this genetically engineered crop, then we can use those technology and modify the genes, and those crops can be released. So there are certain biosafety issues of the genetically engineered crop, but you know that there are many studies have been conducted for multiple years, and in general these crops are safe, it is known. But now our technology, or even that common technology now available, is that to modify this plant's own gene. Now when we modify this plant's own gene using this gene editing technology, so basically that may not be considered as truly genetically engineered. And that's what happened, that now the Indian government has permitted one brassica crop, or oilseed mustard crop, which is genetically engineered or not truly engineered, but is genetically edited. So there is no foreign gene in it. Okay, so those types of crops are allowed. So that's what we are aiming to. So we'll modify plant's own gene, but there will not be any foreign gene or any extra mark will be there as such. So those crops, I think that will be allowed. In fact, all countries are working on this aspect. So if you say that competitors or contemporaries, everybody. So we know the potential of these technologies. So everybody is jumping into it, and so trying to do or use it in some way. For example, you have seen that apple, if you just cut an apple, it will become brown. So if you just using this gene editing technology, if you modify a simple gene in apple, so the apple will not turn brown after cutting. Even that's true for the mushroom. So those genetically edited plants are available in USA. So they have done it. And USA, Europe, they are already doing it, but Europe it is not allowed. But in USA, genetically edited crops are will not be under the purview of the biosafety issues. Let's say that if there is, you can just modify a gene which helps in coloration of this after cutting this apple. So if you just modify those genes, so then apple will not be brown. So in fact, in some cases like mushroom, if it becomes brown, those mushrooms should not be eaten. Okay. So if you prevent that, so then you can actually increasing its nutritional value. So that's one example I have given. But there are many more examples where you can improve even the quality of the crop. Let's say that if we can develop a technology where we identified certain genes which can be modified so that a crop will be resistant to a particular disease. So Indian industry can take those technology and modify useful crops. Like I am working on the tomato plant. So that same idea can be transferred to other crops similar to tomato, like potato. So they can develop another crop like that. So that's what happens actually. So they take the technologies from the laboratories and modify it and use it. So I think it will be useful for Indian industries also. We don't have to hire the technology from outside. So that's the motto.
We heartily thank Professor Kundu for sharing with us his vast knowledge and experience. We also have Professor Gaurav Gangopadhyay with us. We will listen to his vast research experience as he adds on his valuable insight to Dr. Kundu's work.
Let me define a few things. So you are saying about genetically engineered. So if we just go back to history a little bit, the first case of genetically engineered or genetically modified crop, it came to our knowledge when the case of BT. You know, all of us, we today know BT or Bacillus thuringiensis, that particular protein. So it was done in USA. And the maize, so far I remember, it was BT. Maize and cotton was BT resistant. But after that, it was a huge scientific success. But a few years after, the environmentalists were after this because they found death of the insects, because this Bacillus thuringiensis protein is acting against Hemidoptera group of insects, and locusts, their main target. But unfortunately, butterfly, moth, everybody they fall under the same group. So from there, that the apprehension about this genetically engineered or modified crop came. So after that, many countries, they ban that type of research. But gradually, with the advancement of techniques and science, people has now understood that, okay, we can insert something foreign from a bacteria, from anywhere, but first we have to see whether those are stable, whether those have any negative impact on environment or other biological creatures. But nowadays, what we and Professor Pallob, Kundu and our group members, they are doing, they are not trying to bring anything foreign or alien from anywhere. They are just wanting to modify their own genome. So this is just gene editing, what he has told. So we have gene in our genome, but maybe that particular gene is faulty. We can rectify that gene. So you are not bringing anything from outside. With this new technique of CRISPR-Cas and many things, they can minor editing, say A to T or G to C, like this type of tinkering. And with this tinkering, that plant is becoming tolerant. So this is the main thing. Here now that debate doesn't come, and gradually the government officials, bureaucrats, scientific personnel who are under this committee, they are understanding this thing, and they are gradually permitting this genetically edited crop. And before this, what we understand, a rigorous testing is required. So all works, they start in laboratory. In laboratory, we have, say, well-secured tissue culture room. There is one sort of, say, after that it goes to greenhouse. We have greenhouses of many tiers. Say biological safety, we maintain biological safety 1, 2 after that. Because if we want to test this thing, so we have to spray that pathogen, maybe it a paste or a fungal spore. So it cannot go outside the environment. So this is a very critical experiment which we do in our experimental farm where we have dedicated greenhouses. And from that we need a caged field. And I have seen in places in India, in ICRISAT at Patancheru, Hyderabad, where they actually make some nets, and that nets have certain microns pores. Say only that thing can go or out like this thing. So in that consolidated cage, they do this experiment. And after getting the data, they simply destroy the plants because no pollen dispersal can take place. So these are very integrated experiment. It requires permission from competent authority. But this thing we are doing very much because we have all the facility. We have state-of-art laboratory. We have state-of-art greenhouse facility, biological safety rules strictly. And Professor Pallob Kundu, in fact, is the chairman of this committee. We look after this thing, and we believe that with the change in this government policy, we'll be able to deliver these genetically edited crops to our nation very shortly.
We heartily thank Professor for sharing with us his vast knowledge and experience.